Proteins. All right, so protein digestion begins in the stomach. There is no protein digestion in the mouth. We don't have any enzymes for it, and the pH of the mouth is such that most of the proteins would be so globular that we wouldn't be able to utilize them anyway. So protein digestion begins in the stomach with pepsin. Remember, pepsin is released as pepsinogen, and then it's cleaved to pepsin by the acid or by other pepsin. Okay, so it's one of those positive feedback loops. Pepsin works at a very low pH. In fact, if you take pepsin and put it in water, it doesn't work because the pH isn't low enough. You actually have to add acid to make it work. Pepsin takes big proteins and makes them small proteins, right? Fragments. We have oodles of words for protein fragments in medicine. To me, I am not exactly sure what the distinction is between all the words. So I'll give you an example. Proteoses, peptones, and polypeptides are all words for protein fragments, all right? Polypeptides are the smallest, proteoses are the largest, peptones are the middle ones. Okay, but there again, I can't ask you to discriminate between those three, okay? Because number one, this isn't a biochem class, and number two, I can't, so I won't ask you to. And I promise it never came up in all of clinical medicine. Nobody ever asked me what a peptone was or why it was never a part of any disease that I ever knew, okay? So that's pepsin. After the stomach, all right, pepsin stops working because of the low pH, right? Because as soon as the stomach empties into the duodenum, that acid is neutralized, the pH comes up to 7.4, so pepsin will stop working. But the products of pepsin's actions are still in the chyme, right? So what comes out of the stomach, proteoses, peptones, and polypeptides. Now we're in the duodenum, so out of that sphincter of Oddi, we get the pancreatic proteases, and there are multiple ones. Remember, the pancreas is the only organ that has enzymes to digest everything. Okay, all of the other ones need other stuff, but pancreas has it all. Trypsin and chymotrypsin kind of work like pepsin. They take proteins and split them into polypeptides. Carboxypolypeptidase does something very specific. It breaks individual amino acids off the carboxyl end of the protein. So it's a very specific enzyme. It's not as fast, okay, but it does do this little trick of one at a time. Then we have a specific protein digester for elastase. Okay, proelastase is what's created by the pancreas. It changes into elastase, and then elastase digests elastin fibers, which are specific to animal protein, right? Meat, essentially. So we have a specific enzyme for that. Okay, so between these two, what we get is now we don't have big proteins, we have small pieces of proteins and maybe a few individual amino acids from the weirdo carboxypolypeptidase, right? Now, the rest of it, going from peptides to amino acids, again, it's going to happen at the small intestinal brush border, at the enterocytes again, okay? In here, we have the aminopolypeptidase and the dipeptidases. Now, they take little proteins and make 2 and 3 chains of amino acids. So, chains of 2 amino acids or 3 amino acids. That's a dipeptide or a tripeptide. Remember that vocabulary? A peptide is part of a protein. So this is at the brush border. You're not in the cell yet. Now, once the materials, these di and tripeptides, come into the cell, and then the peptidases inside the Bart-Simpson cells then finish the job of going from di and tripeptides into amino acids, single amino acids. This is more important than you might think. You're in an immunology class right now. You've learned all about antigens, right? I'm assuming by now you've talked quite a bit about antigens, right? Well, remember what antigens are made of. Usually they're little pieces of protein, right? If we absorbed chunks of protein instead of individual amino acids, we would be absorbing a bunch of potential antigens. We would be giving our immune system a bunch of things to potentially attack and cause trouble with. So breaking proteins down into individual amino acids before they're absorbed, it helps to prevent immune response to proteins that are foreign to us. You've talked about self versus non-self, right, in immuno. Well, anything we eat is non-self. So if we translocate it, if we move those proteins directly into the blood intact, we'd have a massive immune response. Okay, so this digestion serves multiple purposes. Yes, it creates building blocks that can be made into lots of different things, but it also protects us immunologically as well. Now, there are exceptions, like peanut allergy, for example. Some peanut peptides do translocate intact so that they can generate an immune response. All right, so our final absorbed products from protein digestion, 99% of them are individual amino acids. Whole protein absorption is pretty rare. Then I get to ask you a probing question. Does this match the supplement ads, right? That they're always like, oh, well, this protein, it's going to make you big and buff and make you have great immune system and all that. No, it's not. You're going to make a bunch of amino acids out of it and absorb them and make your own proteins, right? So you might as well pick a much cheaper protein than the very expensive ones they're trying to sell you, right? Because it's all going to get broken down anyway. Okay, so in pictures, stomach, proteins, because of pepsin, become proteoses, peptones, and polypeptides. Then we empty the stomach. Now we're into the first part of the small intestine, the jejunum, right, where we're going to get trypsin, chymotrypsin, carboxypolypeptidase, proelastase. That gets us to small peptides and amino acids. This is at the brush border. And then inside the brush border cells, we have our peptidases that turn what's left into amino acids. That's what's absorbed.